Alright, so this is the second video on enzymes. So I'll just call this Enzymes 2. In the previous video I talked about what enzymes were and kind of how they worked a little bit. Actually, while I'm thinking of it, there is one way that I forgot to mention. When we were talking about induced fit, in fact, let me go back. When we were talking about induced fit, I must have been because we ran out of room on the slide or something, but just as we have this conformational change, the enzyme closes down, so there's a conformational change, the active site adjusts to fit the shape of these substrates, so there's a change in the shape of the enzyme. You can also have a change in the shape of the substrate. So the substrate may likewise adjust to fit the shape. What it can do is bind the substrate can bind the active site, and then there's like a stretching and weakening of the bonds in the process. So as these it might be actually better to talk about the reverse reaction where we're breaking a bond, but in any sense, let's say you have a bond here that you want to break between carbon and oxygen. So there is some vibrational bond. Is not a rigid dowel rod. It's like think of it more like a spring. So it might be like this. It might be kind of stretched out a little bit. Okay, but both in the realm of what the average bond length would be. So what you can have is the enzyme binding, instead of this more stable conformation, once these two bonds are once these two atoms are stretched out a little bit, this bond is lengthened, so you have one part of the enzyme binding this carbon, another part of the enzyme binding this oxygen, but it's binding it in a way that stresses or strains this bond. It's a higher energy. We had just talked about the transition state. If we wanted to break this bond, we can get it up to this higher energy by stabilizing this stretched out bond, and then it's easier to break. A good example of this would be if you've ever tried to, say, cut a shoelace. If you put a shoelace just laying, say, on the table, and you get out your scissors and you go to cut it, chances are the shoelace will just fold up in the scissors, and it might get frayed a little bit, but it's not going to get cut. Right? So that's the example of what's happening here. So how do you get that shoelace to break? Well, you stretch it out a little bit, so you don't just have the shoelace lying on the table. Maybe you're suspending the shoe by holding one end of the shoelace. And you can try this at home. Maybe you don't want to try it with your shoes. Maybe try it with your one of your siblings or something, and hold that shoe up by its shoelace, and then cut it. So you're stretched out that shoelace. You stretch out this bond, and it's a lot easier to cut. So just one other example of how this induced fit model can work. So how are these enzymatic reactions controlled? I already talked a little bit about this with enzyme inhibition. We're going to delve into this in a little bit more detail. Again, an inhibitor is something that causes an enzyme to lose activity, or if you wish, slow down. So what's the point of slowing a reaction down? I mean, aren't these enzymes supposed to speed reactions up? Well, yeah, but if you had a car that could only go 60 miles an hour, it wouldn't be very useful. You need to be able to adjust the speed depending on conditions. So these inhibitors are kind of like the brakes, and they slow it down. Now there's a number of different types of inhibitors. There's, the two main types here they talk about: reversible inhibition. Okay, so this binds the enzyme, but it can bind and then come off, and when it comes off, the enzyme activity is restored. So when the inhibitor is released. So there's two main types of reversible inhibition. So reversible inhibition, we have, and we'll see a model of this in just a moment, but you have an enzyme binding an inhibitor, and then you have an inhibitor enzyme complex, and this either deactivates the enzyme or just slows it down. Right? So causes it to lose activity. Maybe not 100%, but at least slows it down. And then the inhibitor can come off again, and you have the free enzyme back just as you started with, that it's fully active. Okay, so that's what we mean by reversible. You can have the inhibitor bind the enzyme, but then come off and release active enzyme again. Now there's two types. There's competitive and non-competitive. Non-competitive is also known as I don't know why I did it this way, but this is also known as allosteric control, is a non-competitive type of inhibition. So we'll talk about competitive and non-competitive. And then there's another, so two main types again. Call this 1 and 2. Reversible has two types. We'll call it A and B. Competitive or non-competitive, or allosteric control, same thing. And then there's irreversible. This binds to the enzyme and basically destroys it, permanently destroying its activity. So you have an enzyme binding an inhibitor. It goes to an enzyme, and usually this involves a covalent modification. So the enzyme is now covalently bonded to the inhibitor, and now this is a dead enzyme. It is not going to work, and it does not go. Does it go back? No, it's kaput. So we can change the rate, either speed it up or slow it down by these inhibitors or allosteric effectors that I talked about. Sometimes you'll hear allosteric inhibitor. Remember, allosteric just means other site. So allosteric control. It could be an activator, or it can be an inhibitor. It depends. There's some of each. Some speed it up, some slow it down. There's a third type, a way of, and this maybe I shouldn't write this number 3. There's enzyme inhibition. There's also something called zymogen activation. And this is another way to increase enzyme activity. And we'll talk more about how zymogen activation increases enzyme activity later, but it's different from this inhibition. So here's an example of irreversible inhibition. So penicillin is an antibiotic. It binds to an enzyme that's responsible for making so it binds to an enzyme that's responsible for making the cell wall of the bacteria, so it kills bacteria. This enzyme is necessary for making the cell wall, so it binds to this glycopeptide transpeptidase, which is involved in the bacterial cell wall. Not important for this. But the enzyme has in its active site an alcohol group, which was required for function. Penicillin, well, notice here we have, this is a carbonyl bonding to an alcohol, and what you end up with is essentially an ester. Okay, so this covalently bound enzyme inactivates it. You've covalently bound and converted this alcohol group to an ester, and now you've also got the rest of this molecule. And there's probably specific binding sites for these, various functional groups and parts of the drug. So the enzyme binds this tightly and covalently gets modified, and now the enzyme is dead. There's another good example that is that they talk about in that video that I encourage you to watch about SARS-CoV-2 protease, which is required for virus to replicate. There's an enzyme that's part of the viral genome. This protease is made or is encoded by that virus, and it's needed for replication of the viral particles. And this is a target for certain drugs that also irreversibly inhibit or kill the enzyme, that irreversibly inhibit. So watch that video and you'll see, you'll learn more about another type of irreversible inhibition. So as I was saying, there's all sorts of drugs, and these are mostly poisons, cyanide, sarin, parathione, is insecticide, penicillin we just talked about. But so these really aren't drugs, but they do irreversibly inhibit certain enzymes. And the one I wanted to talk about here is sarin here, right? So this is sarin. It binds an enzyme, and I believe, well, actually, DFP diisopropylfluorophosphate is what you see here. So here's an isopropyl group. Here, isopropyl group, isopropyl group. So this is diisopropylfluorophosphate. It's also a nerve gas, but what it does here is there's an enzyme with a serine active site or active site residue, so a serine side chain here. The drug that they talk about in that one video that's used to inhibit that viral protease, it actually has a cysteine. So instead of that protease, they refer to it as a cysteine or a thiol protease. So instead of being a serine, it's actually a cysteine. And instead of an OH, then cysteine has the SH. So when they refer to it as a cysteine protease or as a thiol, remember this is a thiol protease, the active site has a thiol group instead of an alcohol. But anyway, this diisopropyl fluorophosphate, what it does is it bonds here, the fluorine is removed, and we get the carbon-oxygen phosphorus bond. So this here now is covalently modified. So I'll say the drug is, or in this case the poison, is covalently bound to the enzyme. And in that video, they actually show the three-dimensional structures and very specific details, show you what's going on. It's a really cool video. Encourage you to watch it. So that was irreversible, and then we have reversible. And this is a little bit more cartoon model, but it gets the same idea across. You have an enzyme, it binds a substrate, you get the ES complex, the reaction occurs, right? So remember, now it binds the active site. So here's the active site, and the active site is where the substrate binds. Right? It's where the reaction occurs, right? So here the reaction occurs. Substrate is cleaved and it releases products and free enzyme. Now this enzyme can go back and bind more substrate. There's one catalytic cycle. Well, if you have an inhibitor present, it can also bind the active site, but it looks a little bit different than the substrate, right? So I is for inhibitor. It binds the active site, and now if we had, say we had another substrate molecule here, kind of a messed up looking substrate, but it's competing for this. It's kind of like musical chairs. They're both competing for the same active site. So now when the inhibitor is bound, the substrate can't bind. They refer to it as competitive inhibition. Both the substrate and the inhibitor compete for the same active site. So the inhibitor competes with the substrate for the same active site. When that active site is occupied by the inhibitor, the substrate can't bind, and obviously the reaction can occur. Now the thing is, because this inhibitor is binding the same active site, it's generally structurally similar to the substrate. And they show that here, the part that's binding the active site is the same, right? So we can say, this part of the substrate and this part of the inhibitor are going to be similar in structure, because they have to bind the same spot. So one way medicinal chemists or drug makers think about this is, well, we know what the substrate is. We want to inhibit the enzyme. Maybe we don't even know what the enzyme looks like, but we can build a molecule that looks somewhat like that substrate with a few differences and see if that works. Again, this is reversible because the inhibitor, once it binds, it's not covalently modified. It just can come off, just as this is reversible. A good example of this type of competitive inhibition is antifreeze poisoning. So ethylene glycol, I'll draw a structure of ethylene glycol here. So 1,2-ethane diol. That's ethylene glycol. Ethanol is this, WCCH3. Okay, so instead of having this, so this is ethanol, right? Ethylene glycol has 2 alcohol groups instead of just 1. This is very, very toxic. It's found in antifreeze, and it's highly poisonous. So if someone gets ethylene glycol poisoning, actually the enzyme is called alcohol dehydrogenase. If you remember dehydrogenases, they involved NAD converted to NADH. So it was an oxidation. If it was ethanol, it would convert this to ethanal. Right? It oxidizes alcohol to the aldehyde. It's the same enzyme. Well, this gets oxidized by the same enzyme as alcohol dehydrogenase to something that actually ethanal or acetaldehyde is also toxic, but it's not as toxic as the product here. And I'm not exactly sure the product here, so I'm just going to say it gets converted to this toxin. So what we want to do is inhibit this enzyme. And so what you can do is keep the enzyme busy by giving them intravenous ethanol. So the enzyme, this is done in the liver, so the enzyme in the liver is busy, right? This alcohol dehydrogenase is too busy working on the ethanol, and then this stuff doesn't get oxidized to the toxin and just it gets excreted in the urine. So the treatment for ethylene glycol poisoning is one, to pump the stomach, make sure you get all the ethylene glycol out, but then get them drunk and put in ethanol, not too drunk that it kills them, but drunk enough so that the ADH is too busy working on the ethanol and not on the ethylene glycol. Now, one thing that I'm going to talk later about enzyme substrate curves. So what we're looking at here is the rate of the reaction. I'll put here reaction rate. Reaction rate, so the reaction rate's increasing here as a function of substrate concentration, and we get these different curves, something like that. Okay, and we'll talk more about that a little bit later, but this reversible inhibition has a specific effect on these enzyme concentration curves. Another type, again, this is still reversible, is this non-competitive or allosteric regulation. So this is another type of reversible inhibition. So we're not covalently modifying the enzyme and just killing it. We have an inhibitor that binds and comes off. So we have this equilibrium here where it binds and comes off, binds and comes off. All right, but notice the inhibitor is not binding the same site as the active site, right? So the active site is defined as the site where the substrate binds and the reaction occurs, right? Substrate binds, reaction occurs, products are released, releases free enzyme, it can go back, bind more substrate. So here we have the active site. Here we have the allosteric site. Allosteric site, which just means other site, some site other than the active site. What this means is the inhibitor doesn't have to look like the substrate. Right? So obviously they kind of both have this square shape, but it's the pointy end that goes in here. Here's the blunt end. So they might look similar, but they don't have to be. A non-competitive inhibitor has a structure that is much different than the substrate. Yeah, I wouldn't say much different. It can be. It can be much different than the substrate. And later on, we'll actually see a case where the inhibitor is actually the product. So the product here clearly looks like the substrate. So it has a structure that can be much different. So it can also be the product of the reaction. So there's a couple different ways this can happen. The substrate, or sorry, the inhibitor binds the enzyme, and notice the enzyme closes up. So there's a conformational change, right? Just when the substrate binds, that induced fit model, the enzyme closes down on the substrate. Well, here we see the enzyme binding the inhibitor first. This closes up, and now the substrate can't bind. Okay, this is what's referred to as a K-type. A K-type inhibition, and the K-type stands it stands for something that's called a Km. The K stands for Km. This is the Michaelis-Menten constant. I'm going to spell constant with a K. So the M stands for Michaelis-Menten and K for constant. And the Michaelis-Menten constant, we'll talk more about that in a little bit, maybe in the next video, but basically it's a measure of how tight the enzyme binds the substrate. We'll talk more about that later. Another way that it can work is it doesn't affect the rate of this. It doesn't affect the binding of the substrate, but when the enzyme binds the inhibitor, maybe it doesn't close down the substrate, but maybe it, I don't know, let's say here the inhibitor is bound. It doesn't necessarily close this down, but in order for this substrate to be converted, the active site has to close down, right? So there's like a hinge here. Well, maybe when the inhibitor binds here, it allows the substrate to bind fine, but it prevents that hinge from closing down. Okay, that would be referred to as a V-type. Okay, so when the V-type prevents the reaction from occurring, but does not prevent the substrate from binding. The K-type, that's what I mentioned up here, prevents the substrate from binding. Right? Prevents binding of the substrate. So the K, why? Because it's Michaelis-Menten constant, which is a measure of how well the enzyme binds the substrate. So if it has to do with substrate binding, it's the K-type. The V-type has to do with the speed or velocity, right? Prevents the reaction from occurring. It slows the reaction down, slows that catalytic cycle of closing down, reaction occurring, opening back up. So this is, V stands for velocity or speed. So both of these are ways of allosterically regulating the reaction. They're showing it as an inhibitor, but it could also be, I don't know, maybe an activator binds. I'll put here A for activator. And it can either speed up, maybe it helps the enzyme actually bind the substrate, or maybe it allows this hinge region to be more flexible. But in any event, it binds at a different site other than the active site. And it's non-competitive, so they do not compete for the active site. So now we get to zymogen activation. A zymogen, we actually saw this already once. A zymogen is a proenzyme or a proprotein. Proenzyme, proprotein, zymogen, they all mean the same thing. All this means is that some enzymes are synthesized through translation in an inactive form. So remember, proenzymes, proproteins, zymogens, they're inactive. And then once it's cleaved here, so there's an enzyme that will break this, a certain peptidase that breaks the peptide bond. So cleavage here, I'll say here, by a peptidase. Cleaves off this, and now the enzyme is active. And I wish they would have done this in a different way. Maybe here is the active site. Right? So the substrate needs to bind in here. Well, maybe this tail here actually can make it go the other way. Right? And it blocks that active site. Once you cleave, that falls away and reveals the active site. So that's one way in which a enzyme can be formed in an inactive form. The reaction occurs, and now the active site's open. Okay. Now there's a few reasons for doing this. One reason is a lot of enzymes or proteins could be potentially deleterious if it was, once it was made by translation inside the cell, it was active right away. Example of this would be digestive enzymes, right? So if the enzyme if it's a digestive enzyme that's designed to maybe cleave proteins, digest proteins, if it's active as it's made in the cell, it's going to dissolve the cell and kill it. So what we want is this digestive enzyme to be exported out of the cell, and then there's a peptidase in the, I don't know, say in the intestines or wherever, that activates it, and now it can work. Example would be trypsinogen. It's a zymogen, because zymogen's in an ogen. Trypsinogen is cleaved and becomes trypsin, a digestive enzyme. But there's others that are proteins. For example, there's collagen and procollagen. So procollagen, like the collagen in your hair and whatnot, we talked a little bit about that. Procollagen gets converted to collagen. Now, collagen is a structural protein that forms that triple helix. Well, you don't want that triple helix forming inside the cell, right? Because if it's formed inside the cell, again, you're going to clog up the cell with this big insoluble protein. You want it to be exported out of the cell, converted into the active form where it can fold up into that triple helix of collagen and then, work properly. So again, some potentially deleterious proteins or enzymes are avoided by first making them in an inactive form and then active. Also, clotting factors. This actually has more to do with the next bullet point. You don't want clots forming willy-nilly. You want them only in wherever the injury occurs, and you also want it to be done quickly. So one of the other reasons we have zymogens is for a rapid increase in enzyme levels, as opposed to de novo. De novo means from new, so making more enzyme from scratch. Essentially what we're talking about here is going, you got to first transcribe the DNA to messenger RNA, and then the messenger RNA needs to be translated. Does it say here? No, translated into protein, and that takes time. Right? That doesn't happen as quickly as it would to just cut off one bond here, one peptide bond, and all of a sudden get an active enzyme. So you can have a pool of inactive enzyme, and all you need to do is lop off this pro sequence, and all of a sudden it's active. So it's a way of making a lot of active enzyme quickly. It also allows for amplification, as in the example of the clotting cascade. And there is a hyperlink here. It says, what does this have to do with shampoo? Maybe I'll see if I can find that video. But basically, it's like the exponential growth of a virus, right? One person, we can actually do it this way, right? Just as with COVID-19, one person infects two others, and then these two people infect two others, and then these people infect two others, and you get this exponential growth, right? Where you start off with one person, patient 0, and in short order you have a whole bunch of people infected. Well, that's kind of what's going on here with the clotting cascade and that shampoo commercial from, I think, back in the 70s. But what's the analogy here is, here we have an inactive form of an enzyme. Okay, we have I just write an activator that converts it to active. Once it's active, it can activate 2 more, and this guy can activate 2 more. So each one of these, you'll see in the next slide, each one of these is a proteolytic cleavage that converts inactive enzyme to active enzyme. So don't worry about all the details here. This is just an example to kind of show you what's happening. There's a bunch of clotting factors here. Clotting factor 7 is inactive. It gets activated by kininogen or kallikrein. These are proteases, and it converts this factor 7 inactive to factor 7 active. Factor 7 active is also a protease that activates factor 11. Factor 11 is inactive. It gets proteolytic cleaved to factor 11 active, and that's done by this activated factor 7. Now, factor 7 doesn't just work on this guy. He's an enzyme, so he binds substrate, catalyzes the reaction, releases the products, and so it's busy doing that. And as it's doing that, it's making more of this guy that is also a protease. It converts factor 9 inactive to factor 9 active. And so you can see this exponential growth as each one of these keeps pumping out more and more active clotting factors. And eventually we have here prothrombin. It's a proenzyme. We get to thrombin. Thrombin is also a protease, an enzyme that activates fibrinogen. So you see the 2 words here. Prothrombin is a proenzyme. Fibrinogen is a zymogen. They're inactive forms of the enzyme that converted to active. Now, this is an enzyme. Fibrinogen gets converted to fibrin. Fibrin is part of the structural component of the clot. So again, you go from an active structural protein to an active structural protein that forms the cross-linked clot and stops the bleeding, and hopefully you live. But you don't want this to occur willy-nilly, because if you get clots forming all over the place, you can have a stroke and die. So it's a way of storing all these clotting factors in an inactive form, so you have a lot of them that can be activated quickly and stop the bleeding quickly. I should also point out, I won't get into the details here, but that's COVID-2 virus binds a receptor called ACE2 on the cells of lung, kidney, intestines. I'm not sure where else this receptor is expressed, but on the surface, I'll just say it's a lung epithelial cell. This ACE2 receptor is also an enzyme, and ACE2 stands for angiotensin converting enzyme. And so one of the things that we talked about in the chapter on enzymes was, on the chapter on proteins, was this proenzyme business. So you can go back and look there, or you can look online and find lots of videos about ACE2. Now, ACE2 converts angiotensin converts angiotensin 2 to, I won't write the whole word out, but angiotensin 1 through 7. So this guy here is also a protease that converts in this case they're hormones, but peptide hormones that converts from an inactive form to an active form. And they actually have opposite effects. I believe angiotensin 2 and angiotensin 1 through 7 are what we call agonists. They actually have opposite effects, and so you can regulate. It has to do with blood pressure and vasodilation and whatnot, but you can regulate that by converting from one to the other. All right, other factors that affect enzyme activity. You saw this a little bit. I was showing you some graphs. We'll talk more about this in the third lecture.